talk about expansion sequences in 5S RNA. Now, this is a very interesting story to me anyway. We've published this paper back in 1981. And that means that it was published before the first 16S RNA complete sequence was available, which means this was the first insertion sequence discovered in a ribosomal RNA. However, that turns out not to be true because Woese in his infinite wisdom had done a catalog of ribosomal RNA fragments from 5S RNA from a Clostridia that actually had an insert as well. So it's kind of interesting. Now, the way this insert works is what's happening is you have extra material right here, right? And you come around. And what happens if you cleave this out, if you break this off and throw it away, you can just reconnect this U and G, and you have a perfectly normal secondary structure of the 5S RNA, okay? So that's the key thing that you these insertion sequences. Of many of them, you can literally remove them, and you're right back to where you were, and so they're not causing problems in particular. Okay, so this is it's more or less the secondary structure of 5S RNA. There's basically 4.5 helical regions. There's the fifth helix here, which involves the non-standard base pairs. Okay, so let's see how we do here. I erased the message. I managed to push the wrong button. Okay. So this is what you've seen already today, multiple examples of are insertion sequences that have been associated with the large ribosomal RNAs. And what we see here on the top is in fact, the thing growing as you go through a variety of organisms, starting with E. coli and ending up with human. This gradually gets bigger as you keep inserting within the insert, okay? And so so the ribosomal RNA has grown over time. So what we wanted to do was to find out how common this insertion was in 5S RNA and what were some of the properties of this insertion. So we did something which in retrospect was sort of stupid. What we decided to do was to look for more examples. Okay, and so this is an interesting thing you should be warned about. If you probably know this, but you really ought to be aware of it. These databases are not very helpful if you want accurate information. Okay. So there's a database here. I'm sorry to name it specifically, but this particular database is a database of 5S RNAs. 5S RNA sequences, nothing else. It doesn't contain the Halococcus 5S RNA with the insert, which is interesting since the Halococcus 5S RNA was actually probably among the first 10 that were actually even sequenced. But it's not even in the database. In fact, no large 5S RNA is in that particular database. So they just simply have a prejudice against large, they don't like inserts. But we went to other more traditional places like RefSeq, and we looked at 127,000 genomes. We found 157 candidates for possible 5S RNAs with inserts, and then when you go through this, it turns out that a lot of them are junk, and 24 with incorrect gene boundaries. So when people annotate these things, they have a hard time finding where it starts and where it ends, and that kind of thing. And there's some problems like people have like with insertions. Like there was a lot of 5S RNAs just sort of had a duplication in the middle of the 5S RNA. And of course, there were a lot of them in which various portions were just ends, so the sequence was not complete. So I can continue forever. So we ended up creating specialized search tools looking specifically for the sorts of 5S RNAs that we thought were interesting. So in the end, we found a bunch of them, but I guarantee you we have not found them all. Okay, but I would just like to say most of the ones we found are actually real. And so we found 79 examples with 13 to 108 residues as the insert. And so we had put a boundary, I think, of 10. The minimum insert we would consider to be interesting was 10. So the smallest was 13. The largest was, in fact, the Halococcus one that we started with, and the 108 residues, got 79 in the bacteria. And then among the Archaea, we have 10 examples here and 25 bacterial species. So a lot of different organisms have this insertion of 5S RNA. This is kind of a summary. There's one archaeal group, which is the Bacteriales, which includes Halococcus and Halobium and a bunch of things. And then a bunch of mainly anaerobic organisms in the Clostridiales and the Halobiales, however you say that, the Thermobacteriales and the Ultramodales, which is actually a gram-negative group. And we were very upset we hadn't found any gram-negatives until at the very, very end of the search we found this guy here. And so there's a fair number of these 5S RNAs containing insertions. So this is a slide. This has been previewed by the CIA, and they don't want you to get access to this information. So if the print is very, very small, so that if you're not in the first or second row, you can't read it. However, this is a summary. This is a summary of how big the insert is. So here's an organism name over here, which you can't read and you don't care about anyway, and it has an insert of 61 residues. Okay, and these down here, this guy has 32 residues. This one here has 3 inserts in the same organism, has 3 inserts, one of length 29, one of 39, and one of 85, which is sort of interesting. And so we have different sorts of things. Now, there's also, this is a grouping of the organisms that have the large inserts, and it's basically what we have is we have the inserts. I'm not going to show it to you, but we have them categorized into kind of a structural grouping as to where they are and all this kind of thing. And so this is telling you the structural grouping, and we have 17 distinct structural groupings. And then this is showing you which grouping it is, and this is showing you the position, I think, okay, of the thing. Anyway, so there turns out to be then 16 locations in which you find 5S RNA insertions. And the interesting thing here is if you start here at the 5' end and you walk around the 5S RNA, when you come to these helices, the insertion is always on the 3', always on the second half of the helix. Coming around 5' to 3', and as we do the second half of the helix, that's where you get your insertions. And the only exception to that is right here. I don't know if that means anything, but it's kind of strange and interesting. Now, some of these insertions occur with greater frequency, so there seems to be hot spots for insertion in different things. So this is a subset of what was on the previous slide. Now, we have hot spots for insertion, yeah, okay. And some of these hot spots, I mean, we've been looking at the structure of the 5S RNA, and this area right here is one of the most conserved parts of the 5S RNA, so I'm kind of shocked to see, insertions occurring here. We had expected them over here from some prior mutational work that we had done. Okay. Well, I should tell you about 5S RNA in particular. 5S RNA is typically part of an operon. 5S, 16S, 23S5S is a common operon among bacteria and archaea. And when they're frequently in many organisms, there's multiple copies of the operon. So E. coli has something like 6 or 7 copies of the operon, each of which has a 5S RNA. And so in some of the cases, these operons, some of the organisms we looked at, have in fact insertions in more than one copy of the insertion. And one of the more interesting ones, there's an organism which has 6 copies of the 5S RNA gene, 3 with the insertion and 3 without the insertion. Okay. Now, if you only have 1 copy of the 5S RNA, we assume it's functional. If you have 6 copies of the identical 5S RNA, it's probably also still functional. But if there's only 1 copy, we're pretty confident it's going to be functional because 5S RNA is kind of important in most organisms. Anton will disagree with that in a bit. Okay, so sometimes multiple genes exist, but not all have the insert. So this is in the same organism. You see, you got the insert here in this case, and it's not present here. Okay, and so then that raises the question: when there's more than 1 copy, is this really functional? We suspect so, but we haven't done any experiments to indicate that. So here's another case. Sometimes there's more than one example of the intron. And so here's a case where we have in the same organism, right, with two different versions of the 5S RNA, one of which has the, it has the insertion here, right? And this other one has the insertion in the same place as over here, but it has an extra insertion out here, small insertion here. So that's sort of, there's lots of strange things that show up. I should mention to you, I suppose, how is the insertion accommodated in the ribosome? That's sort of a fundamental question. And this is the predicted secondary structure of the Halococcus insert. And we have up here the 5' end of this thing, okay, and it goes all the way around back to the 3' end. And so what we have are two major helices. We have a helix here above the insert, and we have this huge helix here below the insert. Now this is a predicted structure, but it's probably pretty good because you got lots of base pairs. Now, we had a poster downstairs, some of you may have noticed it, in which we actually have been doing some cryo-EM work with collaborators to understand the structure of the 5S RNA and how the insert's accommodated. And it turns out that there's a little, we have like a 7 angstrom resolution structure, and when you look at it, there's two bulges. There's a little, very small bulge, which apparently corresponds to this top part here, and then there's a larger bulge, which appears to, which is probably corresponds to this. But what's likely happening is sort of speculative. We're just saying this has a very extensive secondary structure, so it's pretty much a rod. And so what you can do is if you put that rod in the right place in the ribosome, it'll just go out in the middle of nowhere and cause no problem. So we suspect that's how they get accommodated, by just a little bit of these asymmetrical loops along the way. There's all these asymmetrical loops that allow it to, instead of being a totally straight thing, it's probably bending a little bit, and then it ends up going off in a direction which is not getting anybody's way. And so that's sort of okay. Now, before I talk about this, there's what was I going to say? I forgot, so I won't talk about that. These 5S RNA expansions are actually rare. So, I mean, we found a lot, right? So we found like 75 of them, but the reality is that's 75 out of 100,000 genomes. I mean, they're pretty rare, actually, and only occurring in 4 genera. Okay, I don't know if it means anything, but even the bacterial examples, I mean, are actually halophilic. So not all of them are halophilic, but a lot of the organisms that have this insert are halophilic. There appear to be preferred locations, which is on the hot spots, okay, and frequently or sometimes multiple copies exist, and we suspect most of them are actually functional, okay? And then there's a question which comes up, which is kind of the origins question: why are they mostly recent? Now, when you have examples from the same organism, different species of the same organism, so you got the genus and sort of different species, it's the same organism, they have the 5S RNA, and you compare the sequences, what you find out is there's some sequence differences, and those sequence differences they take care of the structure, okay? They keep the structure valid. And so the question we have is, since then, some of them are old in the sense of 100,000 years, but none of them are old in the sense of the 4 million years that we would like, okay, in some of the earlier talks. And so I don't know why you don't have any old 5S RNA expansions, but we don't. And Jessica, she wants me to answer questions.
Yes. Do we have any questions? Okay.
I was just wondering if there's been any RT-PCR work that demonstrates whether these insertions are actually edited out of the ribosomal RNAs?
Are they edited out? They're simply in mature cells. They're not edited out. I mean, we have an ongoing experiment where we trying to insert them into the 23S RNA, and so they interact with the 23S RNA in a very characteristic place, and we want to we want to lock it in and see if the 5S RNA has to be separate from the 23S RNA. And that experiment is kind of ongoing for the last 5 years.
Yeah, I was just wondering whether there are any cases in which these sequences of RNA are ever translated. I'm sorry, translated into proteins.
They're
not supposed to be, I guess, but
they're not supposed to be. We haven't specifically looked in the case of 5S RNA. People have looked in the case of the large ribosomal RNAs, and there's one area in the 23S RNA that some people are convinced is coding. Even though it's in the 23S RNA gene, it might be coding something. So but there hasn't been anything like that found for 5S RNA. Yeah. The other thing, one thing I didn't mention is I showed you the way the insertion occurs. You cut it out and it goes away and there's no problem. But occasionally, some of them actually, you can't just cut it out and put it back together. You have to do some kind of insertion or deletion event along with it. But the vast majority of them is a simple cut and it goes away. And you just seal it back up.
Thank you.
Am I allowed another one?
We have time.
Michael wants to talk.
I'm sorry. Afterwards. Okay. Thank you, Dr. Fox.
Thank you.